Hello viewers, here is a brief description about digestion of lipid. First of all, come to the dietary lipids. An adult daily consume about 60 to 150 gram of lipid, out of which 90% is triacylglycerol and the remainder consists of primarily phospholipid, free cholesterol, cholesterol ester and free fatty acids. Here are the lists of lipolytic enzymes that are present in GIT and aided in the digestion of lipid. First of all, in saliva, there is lingual lipase, then in gastric juice, there is gastric lipase. Pancreatic juice contains pancreatic lipase, colipase, cholesterol esterase, and phospholipase A2. And finally, in succus entericus, there is enteric lipase or intestinal lipase. Now come to the digestion of lipids. Like carbohydrate, lipid digestion starts from our mouth by the enzyme lingual lipase, which is mainly secreted by the lingual glands of mouth. But the role of lingual lipase is somewhat debatable because we can find some other statement from different sources such as lingual lipase cannot start lipid digestion in mouth because food stays in mouth for a very brief period and the optimum pH for the lingual lipase activity, that is 4 to 4.5, whereas the pH of saliva is near about 7. Then from mouth food enters into the stomach and the action of lingual lipase continues into the stomach. In stomach, both lingual lipase and gastric lipase act together and causes the breakdown of lipid, mainly triacylglycerol, into fatty acid and diacylglycerol. Diacylglycerol means a glycerol molecule containing two fatty acids. From stomach, food finally enters into the small intestine. Rest of the lipid is digested by combined action of bile and pancreatic lipase enzyme. Since the fatty foods are water insoluble in nature, they have a tendency to aggregate it together and to form a large masses. By forming this type of large masses, they reducing the total surface area on which the digestive enzymes could not act properly. For this reason, emulsification is needed. Emulsification is a process by which larger fat globule is broken down into many smaller particles of fat by emulsifying agents. Emulsification done by mainly two complementary mechanisms. One is detergent action of bile salt and another one is mechanical mixing due to peristalsis. First of all, the emulsifying agents of bile, that are bile salts and lecithin, which is a phospholipid, decreases the surface tension of lipid droplet and allows the lipid droplet to be broken down easily by intestinal agitation or intestinal movement. After broken down to smaller particles, bile salt immediately form a thin layer around the small lipid particles and prevent the coalescing of lipid particles, and thereby increases the total surface area of fat droplets on which the water soluble digestive enzymes can act very effectively. Now come to the lipases. We have two types of lipases. One is pancreatic lipase and another one is enteric lipase or intestinal lipase. Pancreatic lipase is the most important enzyme for the digestion of triacylglycerol and present in enormous quantities in pancreatic juice. In addition, enterocytes of small intestine also contains small amount of additional lipases, which is known as enteric lipase. Both pancreatic lipase and enteric lipase are needed for the digestion of triacylglycerol specially. Now, the pancreatic lipase digests triacylglycerol to produce free fatty acids and 2-monoacylglycerol by removing fatty acids from the first and third carbon of the glycerol. Here is the triacylglycerol on which pancreatic lipase acts and produce free fatty acids and 2-monoacylglycerol. Mono means one and acyl means fatty acid. Monoacylglycerol means one glycerol molecule containing a single fatty acid at its second position. That is why it is known as 2-monoacyl glycerol. Here is the triacyl glycerol containing 3 fatty acids and pancreatic lipase removing fatty acids from the first carbon and also from the third carbon. And here are the free fatty acids, and the remaining portion is glycerol containing a single fatty acid at its second position, which is known as 2-monoacyl glycerol. Now, the small amount of 2-monoacyl glycerol isomerizes to 1-monoacyl glycerol, which is then digested by intestinal lipase in the enterocyte to fatty acid and glycerol. In this way, about 25 to 30% of triacyl glycerol is completely digested to fatty acids and glycerols by this process. Therefore, the primary end products of TAG digestion are fatty acids, 2-monoacyl glycerol and glycerol, where 2-monoacyl glycerol represents about 70% and the fatty acid with glycerol represent 30% of the total end product. Most dietary cholesterol remain in the form of cholesterol ester, which is hydrolyzed by pancreatic cholesterol esterase or by the cholesterol ester hydrolase enzyme to free cholesterol and fatty acid. Here is the structure of cholesterol ester and become digested by the enzyme cholesterol esterase and to form free cholesterol and free fatty acids. Finally, dietary phospholipids are digested by phospholipase A2 enzyme, which releases the fatty acids and converted to lysolecithin. Now, after complete digestion of lipid, here are the final end products of lipid digestion. These are free fatty acids, glycerol, 2-monoacyl glycerol, free cholesterol and lysolecithin, which is derived from phospholipid after releasing fatty acids. Okay, that's all. Thank you.